Hello, everyone. It is so great to be able to provide this talk. Thank you so much to Hero for inviting me to give this talk on World Snake Day. My name is Justin Bernstein. I am a postdoctoral fellow at the American Museum of Natural History, and today I will be talking about how over a variety of research projects, we've used molecular data sets like DNA to better understand the evolution, dispersal, and the biodiversity of snakes in Southeast Asia. So after this title slide, I'm going to turn my camera off and we'll get on with the talk. So I want to start off this talk by showing you this photo because it encapsulates a large theme that I'll be touching upon, which is this concept of islands acting as nature's laboratory. So we call islands nature or natural experiments because their isolation acts as a living laboratory where environmental and evolutionary processes occur on a smaller and more visible scale. So we can use islands as a model in order to better understand biodiversity and evolutionary processes. So in this entire photo, we have our mainland, we have really small, tiny islands, we have some larger ones. And not only that, but we also see that we have deep areas or deep waters. And then we also have much shallower waters where you can see that some of these islands, at least above the ocean surface, are actually connected underneath. So this is a theme that we're going to be seeing as we talk about the evolutionary processes in different snake lineages in this presentation. So in calling islands nature's laboratory, there's a lot that we can learn by performing evolutionary studies, especially when we're using molecular data sets on species groups or organismal groups that are distributed across islands. We can better understand how organisms colonize islands and how they're distributed across landscapes. We can better understand extinction processes that are typically higher on islands, and this also relates back into biodiversity as well as conservation studies. And we can learn what geoenvironmental dynamics are playing roles in the evolutionary process that gives rise to new species or speciation. So if we're going to study evolutionary processes across islands, we need a geological system of islands to study. And so a lot of my research takes place in Southeast Asia, and specifically the Indo-Australian archipelago, which is shown right here. So this consists of Indochina, as well as these islands called the Greater Sunda Islands. We have the Indonesian islands, as well as Timor. This is shown in a region called Wallacea. And then we also have the Philippines right here, and then New Guinea, which is north of Australia. And you'll see different terms here like Sahul, Wallacea, and Sundaland. And these actually refer in part to the geological histories of these landscapes. So this entire region actually was very geologically unstable and still is. This map right here shows during the Pleistocene, thousands of years ago, land connectivity. So this is the current geological landscape with gray areas showing land above the ocean surface. But if we go back in time, there were actually sea level fluctuations that exposed land bridges that are now underneath the ocean surface. So what we can see here is that in Sundaland, we have Indochina and the Thai Malay Peninsula connected to the Greater Sundas. So this is where we have the greatest extent of land bridges. In Wallacea, we hardly have any land bridge connections between islands. In the Philippines, we have a moderate amount. And then on the Sahul Shelf, we have the island of New Guinea connected to Australia. And what we know is that different organismal groups in the past took advantage of these land bridges to get from one area to another. And then when these land bridges got cut off, when sea levels rose back up and covered these land connections, our populations got cut off from one another. And this is a way where we can have the evolution of lineages or speciation, the rise of new species. So this is why we call this the Pleistocene species pump hypothesis. With that said, this is also why we use this region as a model region for biogeography, which is going to be using Earth's geological history and knowledge of this to better understand the evolution and the distribution of species on Earth. So a lot of my research is about investigating this biogeography of the Indo-Australian archipelago. And what I've been interested in is trying to identify if these Pleistocene sea level fluctuations that essentially expose these land bridges and then also submerge them back beneath the ocean surface, were these really responsible for generating all of the species or a majority of the biodiversity that we see in the Indo-Australian archipelago? And the reason why I ask this is because this is a very wide region that has a lot of environmental heterogeneity, so a lot of different climates and a lot of different geological histories, and other geological mechanisms that can also give rise to species. So over time, we've really relied on this idea of these sea level fluctuations generating biodiversity, but I want to investigate if in a variety of snake lineages, were there other geological mechanisms responsible for this, or responsible for the species we see today? I also want to know, do environmental factors influence distributions? So here, with geological, I'm talking about the physical landscape, but for environmental factors, I'm really talking about if things like temperature, precipitation, and seasonality are going to impact where we see the species that exist today. The snakes that I have been investigating are in the families Homolopsidae and Colubridae. So the colubrid snakes are represented by Stegonotus and Lycodon. These are also known as ground snakes and wolf snakes. An example of one is shown right here or right here, very charismatic. And then we also have these homolopsid snakes, also known as Asian mud snakes. So these homolopsid snakes are typically found in very muddy and dark aquatic environments, and they are nocturnal. And specifically, these homolopsid snakes are very interesting. It's a small family. Many of them have these rear fangs in which they have a grooved fang to help deliver a mild venom that helps incapacitate their aquatic prey. A few of them have oral glands that likely help them to tolerate brackish and salty water environments. And then we also have very bizarre species like Erpatron tentaculatum that has these mechanoreceptors that are found on the front of the face to help them identify and catch their prey when it swims by. So all of these species are found distributed throughout the Indo-Australian archipelago. So I've studied the evolutionary processes that have given rise to their biodiversity and see how that correlates with these Pleistocene sea level fluctuations. So we use these snake systems in an approach called phylogenomics in order to investigate their macroevolutionary patterns and test these hypotheses of geological mechanisms and environmental mechanisms that can contribute to these biodiversity patterns. So we perform DNA sequencing called genomic sequencing to get thousands of different parts of the genome. And then we use this to reconstruct a phylogeny, which shows us the evolution of these lineages, shown by these lines, as they go from the past into the present. So this branching pattern is similar to a family tree. We then use this as a backbone to estimate divergence times to determine when lineages evolved from one another or diverged from each other, and also use other computer models to better understand ancestral ranges. So what that means is we can identify likely where these snakes were living in the past across a geological landscape like islands. So now we have the evolution of a group. We have information on when the species evolved and where they evolved and where they were living. And with that information, we can test our hypothesis of how different sea level fluctuations or other environmental mechanisms can give rise to biodiversity patterns in the Indo-Australian archipelago. We also perform something called species distribution models. So what this does is we take occurrence data, or where snakes were found, and subject it to a variety of computer models with bioclimatic variables, for example, temperature, precipitation, seasonality. And then we get this, what is called a stacked species distribution model, where we get this heat map, where hotter colors show higher levels of biodiversity, and cooler colors show lower levels of diversity. So what we're seeing here is how biodiversity can correlate with different environmental factors. And so here are some of the results from one of these studies. So this is going to be a phylogeny showing the evolutionary relationships of Stegonotus and Lycodon. So these are these colubrid snakes. And what you're seeing here is you have all these little color nodes, these pie charts. These are those ancestral ranges. So some of what we're seeing in this phylogeny is that these divergence dates, well, these take place before we have those land bridges that were in Southeast Asia being submerged beneath the ocean surface, which means that there's other mechanisms other than these sea level fluctuations that gave rise to the species level biodiversity of these snakes in the Indo-Australian archipelago. So what we see is that a lot of these groups are geographically based, and these colors that I'm showing in these boxes, which pertain or correspond to those ancestral ranges, can actually be shown in the map right here, where we have a group in Indochina, a group primarily in the Philippines, those in Wallacea, as well as New Guinea and Australia. So if we take a look at this map and want to look at this, what we would call this biogeographic story, or how these groups have dispersed over time, we see the following. We see that earlier lineages, and we have our phylogeny shown right here, colored by different groups. We have earlier lineages that were speciating within Indochina. And then we had dispersal moving northward. So this is this group shown right here in blue. And then we had dispersal east and west into South Asia, and then across land bridges eastward into the Greater Sunda Islands. So now we're looking at these yellow and these red groups. We also had dispersal from the Wallachian Islands into the Philippines, and then finally more distributing eastward into New Guinea and Australia, where we had some other dispersal events. So what we actually see here is that we have land bridges that are both deep in time or shallow in time, and also those that were over water dispersals and those that are over land bridges. So you can see those by these arrows shown here. Again, these correspond to the different groups within Stegonotus and Lycodon. And we see that at older timescales, we had this movement over land bridges, where these land bridges were allowing these populations to get from, say, Indochina into Sumatra, Java, as well as Borneo. And then the dashed lines are going to represent overwater dispersals, where when we didn't have land bridges, we have this island hopping, or these rafting events, as we would call them, as they got from the western parts of their distribution into the eastern parts. And from there, we had more movement over land bridges, like going from New Guinea into Australia. Now, something I also want to focus on is what we see in the Philippines, because we actually have a couple of results here that do suggest island hopping, but also other results that warrant further investigations. In the Philippines, what we find is that we likely have island hopping scenarios going from the southern islands into the northern islands. And then we have one species, Deinodon sealii, which is only found in Palawan, likely rafted on what we call this microcontinental block into its current position, which was originally closer to northern China. So all the Philippines populations are more closely related to one another, except this one species. So we have a hypothesis that it either came from this continental block that rafted southward, or it came from Borneo and then dispersed into Palawan. However, these need further testing with additional data. When it comes to these homolopsid snakes, when we look at their phylogeny, we find very similar results. All of their divergent states, when these species evolved, predate those Pleistocene sea level fluctuations. So that means that there's other geological or environmental mechanisms that contributed to their diversification. And what we find is that depending on where in this tree we look, we find that river catchment events, stumbling over my words, tectonic uplift events like the rise of plateaus, or island formation. All of these, depending on where we look in the tree, contributed to the biodiversity of homolopsid snakes. These river catchment events are when rivers will shift north, south, east, or west due to geological forces. And like I said, tectonic uplift events, examples of these are the rise of plateaus, such as the Korat Plateau in Thailand. When it comes to these uplift events, we actually found signatures of undescribed diversity. So we ended up describing a new species of rice paddy snake. And in addition, our results showed that there are bioclimatic or environmental variables that also facilitated the diversification of these snakes. So these are going to be things like temperature and seasonality and precipitation, which I had mentioned before. I don't want to get too far into the details of these computer models, but we can look at the niche overlap of these species. So here we have the distribution of one species in red and then the other one in purple. This KP is the Korat Plateau that led to the divergence of these two species. And we can plot the available niche space in this polygon in purple, and then of that same species, the occupied niche space. We can compare this to the available niche space of the other species and also plot its occupied niche space. And what we find here is that, yeah, we do see a great deal of overlap in the available niche space of these two species. However, the occupied niche space where they are actually found is only overlapping by about 20%. So this shows that they have niche differentiation, and diversification has also been facilitated by these environmental factors. This arrow you saw pop up shows a centroid shift in the niche space as these homolopsids had evolved over time, and we find that things like precipitation was a major contributing factor to the diversification of this group, not just the rising of this plateau. Also, when it comes to the colubrid snakes, we also find that using these environmental variables, we do find that there are limits in distributions, such as at the Isthmus of Accra, that are pertaining to different environmental factors, such as precipitation. So for the sake of time, I'm not going to get too far into this. However, this actually is not in press anymore. This was published this year in Journal of Biogeography. Now, I'm not saying that these sea level changes are not important. We have one species, Hydroblabies periops, that likely use these land bridges to get from Indochina into Borneo and then went extinct on the mainland, or it got into Borneo and then speciated. So these sea level fluctuations, they are important. They do help explain the distribution of species as well as the evolution of others. However, I do think it's time that we start deviating away from this species pump paradigm, or that Pleistocene sea level fluctuation hypothesis, as the only hypothesis. There are so many different ancient geological and environmental factors and events that have given rise to biodiversity patterns within the Indo-Australian archipelago. And so it's time that we start taking more of a brute force approach, or at least testing multiple hypotheses rather than just relying on the one of these sea level fluctuations. With that said, thank you very much for listening. I hope you all enjoyed the presentation. A huge thank you to my collaborators, my funders, as well as all the natural history collections that have loaned specimens over a decade of research for all of these projects. And I would also like to announce that I have just been hired as an assistant professor at Slippery Rock University, and I will be starting the Serpent Lab. So if you are interested in collaborations or want to talk research or other educational opportunities, please reach out to me. Thank you very much and have a great day.